Just a moment. I'm going to take you through this lecture on bacterial genetics. So at the end of the lecture, I hope you'll be able to discuss microbial gene structure and function, define and describe extrachromosomal genetic materials like plasmids and transposons, define different types of mutations, describe and discuss the methods of gene transfer in bacteria, and discuss the process of genetic engineering and its applications.
Excuse me, Doc.
Yes?
We can't see your screen.
Oh. You can't, okay. Sorry about that. Can you see it now?
Yes, Doc, you can continue.
Okay. So I was saying at the end of the lecture, you'll be able to, just a moment. Anyone who knows how to remove this upper panel here? Okay. That's better now?
Yes, Doc.
So we are going to discuss the microbial gene structure and function, define and describe extrachromosomal genetic material. These are the plasmids and transposons. Define different types of mutations, describe and discuss the methods of gene transfer in bacteria, and discuss the process of genetic engineering and the applications. So these are the things we are going to discuss, including the replication of bacteria, what we use bacterial genetics to, for example, doing the preparation of antibiotics and so on. And then in genetic engineering, where we have the gene splicing and so on. So for the introduction, bacteria have ways of passing genes from one set of generations to another. And the unit of inheritance for a bacteria is DNA, unlike the viruses, which will have either the DNA or the RNA. So for the DNA structure, it is composed of nucleoside bases. We have the purines and pyrimidines, guanine and adenine being the purines, and cytosine and thymine being the pyrimidines. The DNA is usually in a structure that forms a double helix arrangement. And DNA in bacteria occurs as a single circular molecule. And sometimes we have small circular molecules that are independent of the chromosome called the plasmids. So this is just a diagram to show you the DNA helical structure, the double helix plus the bases. So for the structure of DNA, we have the deoxyribose and the phosphate residues that are arranged alternatively. And then attached to each deoxyribose sugar are the nitrogen bases, these ones. And as I had mentioned, we have the purines and the pyrimidines. So the adenine is usually attached to the thymidine with a double hydrogen bond, while the guanine attaches to the cytosine with a triple hydrogen bond. And this is the structure that forms now the helical structure on the right here. For the central dogma of molecular biology, it was first articulated by Francis Crick in 1958, and it just involves processes from replication of DNA, transcribing the same DNA into RNA, and then translation of this RNA into protein, our protein of interest. So we have different types of RNA. So we have the messenger RNA, which is like a copy of information of DNA that is brought to the ribose where the information is translated into a protein. So this one picks the information from our gene of interest, and I made a demonstration on your right here just to show that it is copying the information. And then we have the ribosomal RNA, which now is the protein factory of the cell, like this factory here. And then we have the transfer RNA, which now brings the amino acids to the ribosome. It's like transporting the amino acid from where it is being made, then bringing it to the ribosome. So the second objective was the bacterial genome. So for the bacterial genome, we have both chromosomal DNA and extra chromosomal DNA. From this diagram, you can see that on the left here, this is the chromosomal DNA. And then on the right, we have extra chromosomal DNA where we expect the plasmids and the bacteriophages. So plasmids are basically circular molecules of DNA, and they are hundreds of thousands of nucleotides long. They often contain virulence factors that contribute to disease susceptibility. An example of these virulence factors, we have antibiotic resistance and toxins. And in biotechnology, we use these plasmids for gene cloning and recombinant protein production. I will explain in the next slides. Transposons are the other extra chromosomal DNA material. And this from the word transposons, you can see like a trans, meaning it's they are able to be moved. Yeah, they are movable genetic elements in a bacterial cell. So they are able to carry accessory genes such as antibiotic resistant genes. And these ones, they can be used to put a particular gene into the DNA of an organism. Any question up to there before I proceed? If there is no question, I'll proceed to the third objective of our lecture, which was on mutations. So mutations are heritable alterations in the gene sequence. This is whereby there's a change in a gene or a chromosome that brings about phenotypic changes in an organism. So there are different types of mutations. We have spontaneous mutations that arise without exposure to external agents or replication errors. And then we have induced mutations that arise after exposure to mutagens. Then we have silent mutations that have no visible effect on the organism. So for the induced mutations, these are caused by mutagens and carcinogens. An example of this being nitrous acid, fibromouracil, the X-rays, the ultraviolet radiation, the viruses, and mutator bacteriophages. We have different mechanisms of mutations. So we have deletions, we have duplications, we have inversions. This is whereby a single chromosome, there is a mutation in only a single chromosome. Then we have insertion, whereby there is insertion of a particular gene. And then we have translocation, whereby you move from one chromosome to the other. I think you'll have a better view of this when you're learning molecular biology, which goes deeper into the mechanisms. So currently we have most reliable methods of detection of mutations, which utilize molecular techniques to analyze the specific genes that have mutated. So for example, we have this gene expert machine. And then on the left here, we have the sequencing techniques. So the question that arises is mutation a bad or a good thing for bacteria? We are going to see that in the next lecture, in the next slide, sorry. So our fourth objective was on gene transfer. So basically gene transfer, just from the word gene transfer, refers to transfer of genes between bacteria, and they're mainly through three mechanisms. We have the first one being conjugation, whereby we have direct transfer of DNA from one bacterial cell to another. Second one is transduction, whereby you use a bacterial virus or a virus that infects a bacteria called a bacterial phage to transfer DNA between cells. Then we have transformation, whereby naked DNA is taken up from the environment by bacterial cells. So I'm going to explain each and every one of them. So I'll start with conjugation. So conjugation is whereby there is a male cell, usually called the donor cell, or maybe a cell that has the F factor. So it is F factor positive. Then there is a female cell, which is the recipient, that is F factor negative. So what happens here, there is a formation of a sex pilus, and this sex pilus is what will be used to transfer gene from the F positive cell to the F negative cell. And this recipient strain is going to be converted into a donor cell. This has been seen in several instances, including beta-lactamase, transfer of beta-lactamase gene that gives the penicillin resistance in various cells. So this is just a diagram to show us and to make us appreciate what a sex pilus is. So we have the F positive cell here, we have the F negative cell, and we have the sex pilus. So the gene of interest is going to move from the F positive cell to the F negative cell. The second diagram here is just showing us a mating bridge of what exactly happens. Yeah. This is another diagram to help us appreciate the same. So we have the F factor here. It is going through the sex pilus, which is over here. And now it converts the cell into now a donor cell, because now it has the F positive factor. This was seen in an E. coli bacterial cell. The second gene transfer method is transduction, whereby there's transfer of DNA between cells by viruses that infect DNA. So there are two different types of bacteriophages. We have the lytic and the lysogenic phages. So the lytic phages are virulent phages that cause lysis to the host cell, while the lysogenic ones, they are able to infect a cell without necessarily causing any harm. Harm meaning cell lysis or death. So what happens? The bacterial phage will come and attach itself to a bacterial cell, and it will have the gene of interest being transferred from the bacteriophage to the cell. This is a diagram that shows the process. So this is a phage-infected donor. It has all the genes that you want them transferred. Then this is the recipient. So once the two once the bacteriophage attaches itself to the recipient bacterial cell, it is able to transfer the genes from this cell to this other cell, the recipient cell. Before I move, transduction has been seen in several instances whereby there is transfer of toxins from one bacterial cell to another. An example is the group A Streptococcus pyogenes, which transfers the erythrogenic toxin. Another example is the botulinum toxin in Clostridium. We have the cholera toxin in Vibrio cholerae, diphtheria toxin in Corynebacterium diphtheriae. And then we have the Shiga toxin in Shigella. So all those toxins are being transferred through the process of transduction. Transformation is whereby we have a harmful DNA we have a harmful bacteria releasing DNA into the environment. So here we'll have naked DNA being taken up by another cell. So we'll have transfer of this naked DNA to the recipient cell. So when does this transformation happen? Transformation happens during the log phase of a cell, and transformation has been used a lot in genetic engineering. So an example of genes that have been transferred from one cell to another using the transformation mechanism include exotoxins, as seen in Streptococcus pneumoniae, Haemophilus influenza, and Neisseria meningitidis. Yeah, so in a nutshell, we have a donor cell here that will release naked DNA into the environment. Then the recipient cell will take it up and then have it as recombinant DNA here. Yeah. This slide is just showing us the summary of the three processes that we've gone through. Transformation being just the uptake of naked DNA naturally by bacteria. Transduction being transfer of DNA using a bacterial phage, this one. And then conjugation being transfer of DNA through a sex pilus, this one. Yeah. So there is one process called transposition, whereby we have a transposon, which is a transposable element, and they are self-replicating DNA molecules. So what happens, for example, when we have chromosomal DNA, for example, this is the circular chromosomal DNA, we will have a transposon in an initial site. So this transposon will process the gene of interest having the transposon protein. So this protein goes back maybe downstream the chromosomal DNA and targets this. So it acts as a splicing enzyme whereby it will cut this DNA, and then it will be able to now take this transposable element and insert it here, and then there is ligation. Yeah. So this could happen not only on the chromosomal DNA, it could also happen in extra chromosomal DNA. So that can also transfer the genetic material from one cell to another when the other processes happen. So recombination is what happens after the gene transfer. So we will have rearrangement and integration of donor and recipient genomes, and this will result to formation of new hybrid genome. So if you want to detect if the specific location where the recombination has successfully happened, then we'll use the genetic mapping methods that uses labeled probes. So after the recombination, it's now when we can have this in it's the process of recombination and gene transfer that now brings us to the fifth objective of the lecture, which is genetic engineering. So genetic engineering is also called gene splicing, or maybe genetic modification, or recombinant DNA. So basically it's just transfer of DNA. So you take DNA from one organism. It could be a cat that is able to, maybe a cat that has a gene of fluorescing at night, and then you add it to another DNA of another organism, like a jellyfish. And what you have is recombinant DNA. And how this happens is because the genetic code is universal in all the organisms. So be it animals or different animals or even plants, we have the same genetic code. We still have the A, C, T, and G. And here we need enzymes, the restriction enzymes, which work as scissors to cut off, and then so that we can have the space to put our DNA, and then we have the ligase that glues it up. So I'll explain through that process in this diagram. So we have DNA1, which is of our interest, and then we have DNA2. So here we'll use a restriction enzyme that recognizes this sequence. So the same restriction enzyme is the one that we will use to cut our DNA to here, because now it is still recognizing the same sequence. An example of a restriction enzyme is ECOR-R1, which cleaves using this particular sequence. So once that is cut, it's when now we will join the two DNA materials, and then we will get them together using the ligase enzyme. And this results into a recombinant DNA molecule. Yeah. So this recombinant DNA process is not complete unless we put this now back into a bacterial cell. So for example, if this was our DNA fragment that we cut using an endonuclease, and then we placed it in a plasmid, for example, and then we have our recombinant vector using a DNA ligase, we have to insert this recombinant DNA into a bacteria. And the reason we are doing this is because a bacterial cell is able to undergo cell division, and in a few days we are able to have a lot of bacterial cells harboring our gene of interest. So what are the applications of this genetic engineering? We use it mostly in diagnosis, research, in agriculture, all those places. So in diagnostic tests, we have specific gene probes that are used to identify pathogens. And then we have the DNA sequencing and molecular typing of microorganisms. And we use this in epidemiological studies. For therapeutic uses, for example, insulin, we are able to manufacture it using the same genetic engineering method. So in insulin is whereby you have the gene of interest, which is insulin. You put it, you get it, do the plasmid process, put it in a bacterial cell, and then you're able to have several copies of that insulin gene. Other applications are apply on social impact, whereby we have human and animal experimentation, ecological disruption. In agriculture, we are able to have genetically modified organisms. For example, we are able to manufacture plants that are able to resist things like drought and so on. But this genetic engineering can also be applied as a biological welfare, and therefore it's a threat in this case. So this is a slide to just give a summary of all the things you can do in genetic engineering, from using it in forensic applications, diagnosing human genetic disorders, in human gene therapy, and all this. Yeah. So it has a lot of applications. And this slide is just a cartoon diagram to show us what exactly happens in gene transfer between bacteria. So this is a cartoon, which maybe harbors the penicillin resistant gene. So it is telling this other one whether it wants to be a superbug. And if it sticks that into its genome, then even penicillin won't be able to have it, to harm it. Yeah. So as we come to the end of the lecture, I have a few questions and I would like anyone to participate. So which method of gene transfer involves direct contact between the bacteria? That's the first question. Anyone from the class can answer?
Conjugation.
Yes. Then transduction is the transfer of DNA through?
Bacteriophage.
Yes. So I believe you've understood the short lecture. If there is any question, then I'm ready to answer. But basically, we've covered the gene structure, the extra chromosomal genetic material. We've covered the mutations. We've covered the methods of gene transfer, and then the genetic engineering and its applications. Thank you.
So allow me to talk. Thank you so much, Dolores. That was an excellent explainer on really the background to genetics, and also in particular bacterial genetics. And it forms a background onto how bacteria reproduce, and also how they pass their genetical material, and most importantly to us, their mutations. We need to be able to know how they move about and how these mutations occur, because this is the beginning of what you're calling antimicrobial resistance. I think you saw last week there was something in the news articles and a big publication on Lancet where it mentioned that AMR, that's antimicrobial resistance, is the biggest killer in the world at the moment. It surpassed infectious diseases. It's even surpassed even as we're talking COVID-19. So really this is our beginning of learning and understanding, and also hopefully we'll be able to get ahead of how we can prevent antimicrobial resistance. I know you've already had some lectures on this, and if not, this will be coming up. And also the last slide where we had the questions, this is just a taste of how your examinations may come, or an example of an MCQ, 2 MCQs, and we just want to see or test your understanding, and also to give you that taste of what is ahead in terms of your examinations. So we welcome any questions, concerns, feedback from you. And thank you. I'm expecting to hear a question or a concern about mRNA and mRNA technology. Hopefully that's something also Dolores can touch on. And there's a question from Adam. Dolores, there's some questions in the chat.
Okay. Oops. Okay, I can see the first question is on, oh, I don't know why my screen is acting up. The first question is the difference between transposons and plasmids. Okay, that's a good question because it always brings confusion. So a transposon is an element that can be transferred from one gene to the other, while they are both extra chromosomal DNA materials in a bacterial cell. However, a transposon is just it's usually on a DNA, and it can be transferred from one particular gene location to another. While a plasmid is usually it's an extra chromosomal, it's circular. And yes, it can also be transferred, but the way it is transferred from one place to one bacterial cell to another is slightly different. Is recombination the same mechanism in which HIV integrates into our cells? Yes, it is the same process. The next question is, when gene transfer occurs, does the gene integrate into the chromosomal DNA or extra chromosomal DNA of recipient bacteria? I think it all depends with what is being transferred and the process. So for example, the most of the things, or rather the technologies we've done in genetic engineering, we are using plasmids, which is an extra chromosomal DNA. So the transfer can happen in an extra chromosomal DNA. And for example, where the transposons are involved, we can transfer genetic material in the chromosomal DNA as well. Yeah.
Everything you said.
Any question I've missed?
Amekadararu na bado hablet kama gari imewachilia breki ama nyari zitafanya nini breki. Aya, naweza kuja sasa hivi.
Hello, can you hear me?
Yes, there's one. I think there are two more. There's one from Mohammed, where are there so many ethical concerns with genetical engineering? It's a good question.
Yeah, that's a great question. So this the ethical concerns is whereby we can use this genetic engineering to mix different DNA materials of different organisms. And what will we have in the end? Because we won't have the original organisms, just the mixed up. Yeah.
And also, I think there's quite a lot of regulation, just to add on to what you're saying, because of that interfering with the genetics or DNA, and that raises a lot of concern even through the legislation, through law. So for us, even as we conduct our research, our experiments, and our work, we also have to pass through ethical evaluation. For us, we have KNHUN board that looks at to how, first of all, we are protecting the rights of the patient. Are we interfering with any of their even genetical material? And if we are going to study their DNA, are they aware? And what are we going to do with their data? Is there privacy? Are we protecting their data as well? We've had cases where people want to do paternity tests. Again, it all goes to looking into the genes. And also now genetic engineering in this, there's always this big debate and point to have any GMO, but now we are seeing research as showing that actually they're quite safe. The other day I saw there's genetically engineered meat, so we don't have to actually kill animals. We can genetically have meat modified from different structures, and we can have that. So the world is changing, and this lecture actually just gives us a good baseline on what it involves in terms of even the next generation of medicine, vaccines, and preventative strategies. So for me personally, I look at genetical engineering as a marvel of science that can actually be used for the good in terms of preventing diseases and also protecting our environment. And I'm sure you can all agree, and also our lecturer for today can agree. So someone asked, what's wrong with that? Interfering with the genetic information of an organism, if it's meant for the good. Yes, it's meant for the good, and there's nothing wrong with it. What is wrong is doing it without, particularly we're doing it in terms of a patient or a human. They should be fully aware, fully understand what's going on, and they have the right to say no before we do any of that experiment or intervention. So it's just making sure that all members or everyone in the public is aware. And Dolores, you can add on to that.
Yeah, I think also the fear is on losing the originality of what organisms have been having. So if you do a lot of genetic engineering on organisms, we might have a loss on that. So that's why it's supposed to be regulated.
Dolores, there's a question here.
What's the question?
Difference between plasmids and transponents.
Transposon.
Yeah, sorry, yes, sorry, I was pronouncing it wrong. Sorry.
Let me just get back to the slide. So as we had seen earlier, the plasmids are just extra chromosomal DNA. So we have the main DNA in a bacterial genome, and then we have the plasmid being the extra chromosomal DNA. However, for transposons can actually be here in the bacterial DNA itself, just a segment whereby it has a sequence that can, a transposable element that can be transferred from that particular place. Let me say, for example, it can be transferred from this particular site and maybe be placed over here, or maybe can be transferred from this part to now the plasmid DNA. That's the only difference. Let me show you a diagram of the transposon that you can appreciate. Yeah. So for example, this one, you see this circular this is a circular chromosomal DNA. So we have this particular site whereby we have the transposon. So this transposon, because it is an element that can be transferred from one place to another in a bacterial genome, maybe it can be transferred from this particular part to this site here, which is downstream the chromosomal DNA, or it can be transferred from this particular place to maybe even the plasmid DNA, the extra chromosomal DNA. Is that clear now? Hello?
That's clear now.
Okay.
Mutation is illegal, yet it is a potential prevention for HIV.
Could you repeat?
Why is it that Delta 32 mutation, I guess it's in the HIV illegal, yet it is a potential prevention for HIV?
You're talking about Delta 32 mutation in HIV, right? Hello? Maybe that. Hello? Can you hear me?
Yes, we can hear you. Yes.
Yeah, I wanted to talk about the Delta 32 mutation. So there are people who are generally, or rather naturally born with that deletion, and they've been found to have a resistance to HIV infection. So yes, we could be using that as we could use that to maybe modify other individuals to also have a resistance to HIV, but how ethical is that? So I think the reason it is not allowed is because of the ethical concerns that come in it. Because now it means you'll be modifying an individual completely. Yeah. Any other question? Any clarification? Dr. Marianne, are you there? Having no any other question or comment, I think we can come to an end of that lecture.
Thank you and good day.
You too. Have a good day.